Hello, everybody.
Yeah, so the question that I had for you was, so given many of the recent advances in protein structure determination with the advances that DeepMind has come up with, especially more recently, I was wondering if you could comment maybe on the role of NMR going forward in the future for in biophysics, and also what are the techniques that you think are interested or of high relevance today?
Well, when we look into the future, we certainly need to put NMR in perspective with other available technology. And the most recent breakthrough has been with AlphaFold and similar IT predictions of protein structures and to some extent nucleic acid structures. The results from these modeling exercises are certainly good enough to start a structure determination in crystals by molecular replacement. And these predicted structures, of course, help greatly to solve the structures by the other techniques, not solution NMR, but by crystallography and cryo-EM. What AlphaFold does not do, and will probably not do for quite a few years to come, is to deal with the behavior of intrinsically unfolded proteins and with any parts of proteins that are temporarily or continuously structurally disordered. So I see quite an important continuation of our work in complementing structures obtained by the combination of artificial intelligence-based structure predictions, cryo-EM, and X-ray crystallography.
Thank you very much for that. I had a related question from one of the other organizers, and I think she was wondering more specifically on what do you think the role of the NMR in combination of cryo-EM, which you mentioned, would be in terms of for the future of biomolecular NMR.
Well, we will continue to solve structures of small proteins. There is no doubt. There will continue to be physiologically important proteins that will, for one technical reason or other, not be amenable to methods that do not work in solution, or where other methods may not give meaningful results. I think that's so. I see full complementarity between all the methods that are available for structural characterization of biological macromolecules.
All right. So we have a question from Nino Wili. He asks, when you started out your independent research, was structure determination by NMR already your ultimate goal? And if not, what did you have in mind?
Well, as you mentioned, I was working with metal ions when I was in Berkeley as a postdoc, and I was also working with metal ions during my PhD in Basel using electron spin resonance for studies of paramagnetic metal lines. So when I went to Bell Labs, I all of a sudden was confronted with having to work with proteins. Now for me, proteins were metal lines surrounded by a bulk of uninteresting material. And so my first studies at Bell Labs, the first relevant studies, were all focused on the metal center in hemoproteins. That is cytochrome C, hemoglobin, myoglobin. And there was a very special reason why I got into this. You see, I was, as you also mentioned, I was mostly in sports rather than in science in my early years. And I was playing soccer competitively, and I was also running middle distances, 1500 meters in particular, and some 2-3 kilometers. And I always ran into oxygen debt. That means the limiting factor in my performance was very clearly the oxygen uptake and oxygen transfer to all parts of the body. So the first protein I actually studied was my own hemoglobin. So I wanted to find out whether I could possibly make some discovery that might help to overcome the so-called oxygen debt in middle. It's mostly in middle distance running, of course in long distance running as well, but there it is not as acute as in 800 and 1500 meters. And so that's how I got into proteins. Then I had to learn the structures of the 20 amino acids around the metal lines, and from there we went on. I went on. I was alone at that time, and I must say that very early on in 1968, I managed to discover a serious error in a crystal structure of a protein in cytochrome C. They missed to properly identify one of the axial ligands of the heme iron. And from that point on, I was sure we could do it. We could get a structure by NMR, except that the ideas I had at that time were useless. And in the mid-1970s, we then got the right idea on how to do it.
Yeah, that's a remarkable story, and I think it says a lot to many of the young people we have today here in the audience. Just because that one should not be, afraid to venture into something new if they're committed and excited about it. So I think we have a question from Gaurav. What topic in NMR can get a Nobel Prize?
Today?
Today, yes.
Well, there has been no Nobel Prize in solid state NMR, and there has been no Nobel Prize in functional MRI. So latter is sort of surprising because there were only 2 participants in the imaging prize. And there could have been a third participant for functional MRI. These are potential prizes that have not been considered. I'm sure that in case that high resolution experiments can be reliably performed in living cells. I mean, seeing proteins, protein interactions, in possibly unraveling mechanisms of control in living organisms. I could see many chances for future Nobel Prizes.
All of those sound like really promising fields. But so Gaurav has a follow-up question. Do you think that hyperpolarization of spins in solution NMR can have an impact on structural elucidation? And is hyperpolarization in solution feasible?
Well, hyperpolarization. Well, no. Hyperpolarization is a very wide field. I mean, this started really in the 1950s. I think the Overhauser paper on solid state NMR EPR in 1953 was the first such experiment. But there have been experiments performed in solution ever since the 1970s. That's not a question. That definitely can work.
Yep. Yes. So maybe with that, we'll go to the other questions. So Francisco has a question asking, what do you think about the 3D structure determination directly in a cellular context?
I didn't understand the question. I'm sorry.
What do you think about 3D structure determination in a cellular context, like within cells?
Well, that's what I was referring to just 2 minutes ago. I mean, in cell, NMR has made progress. It is difficult. It may be difficult to get high resolution structures, but where I see really interesting potentialities is for the investigation of intermolecular interactions in living cells and studies of control mechanisms.
So we have a question from Casper. So as a beginner within the NMR field, I'm curious to know what drew you to the field of NMR and what personal motivations guided your scientific interests?
The question is, why do I do NMR?
Yes. What brought you to the field of NMR?
Well, I started out doing EPR, as you have mentioned right at the start. And I started to do EPR because by chance a machine, a brand new machine, was standing idle in a room at the University of Basel. And I had actually 100% use of this machine once I got it to run. And I just had to get liquid helium from someone, I had to get quartz tubes and so on. And then I got to a stage where I realized that some experiments that Manfred Eigen had performed could be done much more nicely with NMR. Not what Eigen has done, this happened at around 1963. Eigen had studied fast reactions such as water exchange from metal ions and hydrogen transfer reactions by the use of quench methods. So he would very rapidly change the temperature in a solution and investigate the relaxation to the new equilibrium. The same with pressure jump experiments. And so he was able to measure, in particular, water exchange rates from metal ions. And these were in the range, what he could measure was in the range from about 10 to the 9 to 10 to the 6 per second. And the slower exchange he would not see usually. And then I discovered in connection with EPR, that such measurements could be done in equilibrium by NMR. And at that point, I looked in the literature, was there someone who was doing this? And there were two groups that were doing this. It was Taube at Stanford and Koenig in Berkeley. And so I wrote to Koenig and asked him if I could join him and complement EPR studies with NMR. And that's what happened. And I would then, neither could work alone. I needed to run in parallel the NMR experiments that I had developed in Basel, and then use 017 NMR, deuterium NMR, proton NMR, and all this over a time temperature range from about 4 to 160 degrees. So the complete study of the kinetics of exchange reactions, deciding on whether there was hydrolysis or whether water molecules were exchanging as intact entities, and so on. And so that's how I got into NMR. That was really physics. It was physics. You see, we would build our own apparatus. We would prepare coils for 017. This was all relatively primitive, of course, but it got me the job at Bell Labs. You cannot ask for more. So
yeah, that's amazing to hear. So one of our co-organizers, Adrian Draney, he has a question. So early career scientists are often plagued with doubt and where to put their focus. The recipe of success seems to be a mix of grit, good ideas, and serendipity. What do you think is the proper distribution of these ingredients? Would you add anything to this list?
I can only add that uncertainty and doubts are the same for old scientists and for young scientists. As long as you are in research and you are really going ahead and do something new, you really essentially have to wait for others to confirm that you were not wrong. And that's the same when you are 80 years old or 25. Except that you don't have so much time to wait for the others to confirm that you are right.
Yeah, I hope that answers your question, Adrian. Feel free to chime in if you have any other follow-up questions. So we have another question from an attendee, Sriram Raghavan. And he asks, it is always inspiring to study structure-function relationships, and he is working on understanding reaction dynamics of proteins. And he asks if there is a possibility to study allosteric regulation of proteins using NMR apart from MD simulations. And he says that these interactions are very noisy, and can you share some insights?
Well, if I understand the question right, then your colleague inquires about the possibilities of combining information from MD simulations and NMR to characterize ligand interactions with macromolecules. Is that what is at stake here?
Yes, I think so. Yep.
Yeah, then, I mean, we are again in a field where artificial intelligence-based information is still difficult to obtain. So both experimental studies with solution NMR and and MD simulations can do an important job in complementing structural data that show where the ligands sit in the macromolecules, where the binding sites are, and so on. You get kinetic data, you get thermodynamic data, and of course NMR is the more direct way to get it.
Yeah, I hope that answers Sriram's question. So we have a question from an anonymous attendee, which is going back to this point that we had initially about AlphaFold again. So would you see a combination of AlphaFold prediction together with automatic NMR assignment and structure calculation as a more routine approach?
Now I'm again uncertain. Does the question actually ask, can structure predictions help us with solving protein structures by NMR? Is that?
I think the question is more that, like, combining the prediction with automated NMR assignments.
Oh yeah, that's the same thing. That's I understood this well. The question is, does knowledge of a structural model of the quality that can be obtained with AlphaFold, can this make structure determination by NMR more efficient? And I think, in my opinion, it is not as direct as in crystallography. So this combination is most direct. You just start molecular replacement. And if you can get crystals and if you can get good diffraction data, and then you have a model from AlphaFold, you just start solving the structure with molecular replacement. As far as I can see, there is no such direct way to support a de novo structure determination by NMR with structural model. It can help you to be more assured of what you find, but it's not comparable to the help that model from artificial intelligence can support a structure determination and structure refinement, either by crystallography or by cryo-EM.
Okay, thanks for sharing your thoughts on that. We have another question from an anonymous attendee, which seems to be of high interest. So students find that learning curve for NMR is very steep compared to other techniques. Do you think NMR education needs a change?
Well, it is one of the weaknesses of NMR that it requires at least some basic physics to be. There has to be someone with a certain amount of knowledge in experimental and theoretical physics for things to turn out well. There are a lot. We should not forget that there is a tremendous amount of routine applications of NMR that do not require such help. That's very clear. I mean, let's not forget that NMR is probably the most widely used analytical technique in chemistry, in particular in synthetic organic chemistry. Many questions in biology can be investigated with relatively modest knowledge about the basics of NMR, MRI, magnetic resonance imaging in the hospitals has long become a routine thing, essentially push button thing, except that a lot of knowledge is then, and experience is then needed to analyze the results, of course. But then in solution NMR and even more in solid state NMR, even the optimal measurement of experimental data requires a sound background in physics. Simple as that.
Okay.
This has held up the field, of course, compared to crystallography, which is now almost a push button thing. Whereas NMR, I mean, for scientists, it's a nice thing. It is still interesting. It is not a routine thing to study complex biological systems with solution NMR.
Maybe there is also a related question to that last comment. Do you see prospects of how solid state NMR of proteins can influence the liquid state NMR field?
Well, when you look at recent literature, then you will see that solid state NMR is more often brought in connection with cryo-EM than with solution NMR. There are features in solution NMR and solid state NMR which are closely related, but it just strikes me that there is a stronger trend for solid state NMR spectroscopists to compare their data and to combine their data with cryo-electron microscopy.
Okay, yeah, I hope answers the question. And so we have another question from a co-organizer, Michael Hope. So he asks that he's curious to hear the story of like how when you found out when you were awarded the Nobel Prize and where you were at that time, etc.
Well, I was teaching when they called, and my assistant had strict order not to disturb me when I teach. And though they had to call her 3 times before she came to the lecture hall and called me out. Actually, the procedure went, in between they called someone else with my name because they thought they were at the wrong place when she told them that there was no way I could be contacted now. So, and then it was just 5 minutes before they went to press when I learned about it. And so I did stop teaching, and I fortunately had formal clothes in my office because I had to go and give the opening lecture for a structural genomics congress in Berlin the next day. And so I could change, and then the house was full of reporters. That's how it goes. More exciting is that when I went to Berlin in the evening to join this congress, 2 of the engines of the 4-engine airplane burned. There was a fire. We had to make an emergency landing in Leipzig, and then I was brought to Berlin in a car, very comfortable car, I can say. And I arrived there at only, I think, at 3 a.m. And by that time, they had put the flowers and the champagne in my hotel room, which smelled like a morgue because of the flowers. That's how it went.
Yeah, I mean, I think there is a related question too. So maybe for you, is that your most exciting moment, or is there something else that you cherish as your most exciting scientific moment?
Which moment?
Sorry?
Which moment?
Your most exciting, like experimental or your scientific moment, like where you might have realized that.
There were a few. You see, there were a few. Like the experience in '68 to have just clearly corrected the crystal structure of a protein. That was not bad. And well, I mean, to solve the structure was, of course, a greater moment than getting the prize. I took a long sabbatical when I realized we got it. I went skiing for 2 seasons. Basically, I'm a ski instructor by profession. So I left and I went skiing again from 1984 to 86, 2 seasons.
That's just fantastic.
Well, I mean, to solve the structure of the prion protein at the height of the mad cow crisis. You see, well, there were also more reporters then at the press conference than for the Nobel Prize. You see, that was more important. You are too young, you don't remember this. But in England, they were culling 100,000 of cattle at that time, and then the disease swept over into Europe, France, Switzerland, Germany, and to find the structure of the protein that did it was a tremendous thing. And then Draney was also a big thing. See, I mean, these were the big moments, and there are still a couple after the Nobel Prize.
Yeah, I mean, all of those sound amazing. Maybe as we're kind of nearing towards the end of the time that we had requested with you, maybe as one final question, do you have any thoughts or advice that you would like to give to the NMR community in general, and more particularly to the students and postdocs who are in the audience today?
The important thing is that you enjoy what you are doing. And if you have decided on something that you enjoy, then put in all that you have to pursue it. If you don't like what you do, then you cannot fail, because then it's wasted time. If you like what you do, you can afford to fail and fail and fail again. It's not lost time because you enjoyed it. And anyway, you have to make a lot of mistakes to come to a big breakthrough.